Welcome to the class on bird color. My name is Anand Krishnan and I'm an assistant professor at the Indian Institute of Science Education and Research in Bhopal. So I'm going to be teaching you today about bird coloration in today's lecture. And this lecture is going to be done primarily on the chalkboard. As you can see, I'm going to be writing on the board and I'm going to be taking you through an outline of the lecture as I give it. And this lecture will require you, therefore, to follow what it is I'm saying, to make notes for the same, and then refer back to the books. If you want to know about the reference books, you can get them from your TAs. As of now, I'm going to begin by asking you the question, what does bird color actually mean? Color in general refers to what wavelengths of light we get back to our eyes when something light reflects off of a surface. So if light reflects off of something that we see as red, that means it is absorbing all other wavelengths of light and reflecting only red light back to our eyes. Alright? So, as a result, the object appears red. Now, as you know, human beings have three different color channels. The blue or short wavelength, the green or medium wavelength, and the red or long wavelength colors. Okay? Birds, on the other hand, differ from us in actually possessing four color channels. They are what you call tetrachromatic. Okay? And to illustrate that, I will point you to this graph here. This graph on the y-axis indicates the percentage of light absorbed by the photoreceptor, and the x-axis indicates wavelength. So birds have a violet or ultraviolet receptor, a blue, a green, and a red or long wavelength receptor. And these receptors have slightly overlapping but mostly non-overlapping sensitivities. As a result, if we can see a number of different shades by the combinations of our three color channels, just imagine what diversity of colors a bird can see with its photoreceptors. And as a result of this, birds have evolved a diverse array of colors, right? So that leads us to the first problem. How do you actually measure color, right? And in particular, you want to measure color as the bird would see it, not as you would see it. Most common places use what is called a spectrophotometer. So you have a fiber optic probe, you shine light onto a feather and include UV because birds can see UV. And you measure the percentage of light that's reflected back. So if you were looking at reflectance from a blue feather, you get a peak somewhere around here. So imagine this is an entirely ultraviolet feather, you'll also get reflections like this, right? So the photoreceptor absorbs the most light at that wavelength, and a feather of that color also reflects the most light at that wavelength. And the same goes for blues, greens, reds, and all the possible combinations of these that you can get. Okay? Now, colors in birds are due to a number of factors, right? And birds, as you know, span the gamut from blues and violet colors all the way to bright oranges and bright reds across the spectrum. So what causes birds to actually have these colors? Well, we're going to discuss that in this lecture, the diverse ways in which birds have evolved these amazing colors. And most notably, I'm going to tell you right at the outset, when most of us think color, we think there is a pigment that causes color. Our blood is red because of a pigment, hemoglobin. That is not always the case for birds. As we're going to see, a lot of the shorter wavelength colors that birds exhibit are a result of completely different phenomena. But birds do have pigments. In fact, a number of colors in birds are due to pigments, and we're going to discuss some of the major classes of that. Now, how might you identify a pigment? Well, first of all, you have to match the feather and extract the pigment. Once you extract the pigment, you can use a number of tools. You can use high performance liquid chromatography, HPLC, and more polar compounds will elute first. So, You can identify whether your pigment is polar or non-polar, and then you can identify what the structure of that compound is using a number of techniques: mass spectrometry, nuclear magnetic resonance, and Raman spectroscopy even. Raman is being more recently used to identify pigments in fossil feathers from way back. So it actually has a lot of use. It was also most recently, many of you from India will know the name of the pink-headed duck. It was recently used to identify the compound that caused the pink in the head of the pink-headed duck. So these techniques can give you a lot of information, and thanks to those, we've identified the major classes of pigments that make up bird feathers. And because of this, we know that birds that have bright yellow, bright oranges, reds, and pinks are more or less always colored by a group of compounds called carotenoids. Okay? Now, I don't expect you to remember this, but carotenoids are chemically what you'd call 40 carbon tetraterpenoids. Okay? That is what they are structurally. They form a number of different classes. So you can have non-polar substances like beta carotene, which many of you probably know, and polar carotenoids like the xanthophylls. They're polar because they have a hydroxyl group or a keto group attached to them. And that's all you need to know. The most important thing you need to know about carotenoids are twofold. One, they are not made directly by the bird. They have to be obtained from the diet. This is true of many species, including flamingos. Many of you will have heard that flamingos in captivity lose their pink colors. That is because they don't have a dietary source by which to put carotenoids in there, right? The other factor in carotenoids being dietary is that they cannot form patterns. So feathers are either uniformly colored with them or not at all. Remember these two things. If you were to feed a bird carotenoids, it will put them into its feathers. If you don't feed a bird a source of carotenoids, it will not be able to put them into its feathers, and the feathers will become white and lose the color. That is very important to remember. This has been shown by a number of studies. Now, what are these dietary sources of carotenoids? They can be insects, they can be fruits, they can be leaves and other plant material, and sometimes they can be extremely bizarre. The Egyptian vulture, its yellow face is because of carotenoids, and it gets these from dung. So the Egyptian vulture actually eats animal dung, and it has been shown to assimilate carotenoids through there. Now, typically, you don't get carotenoids in their pigment form. It's not that you just eat the pigment and put it into your feathers and you are orange or pink or whatever it is. Most commonly, you get these pigments as precursors. The most common precursors are called lutein and zeaxanthin. This is just for interest, you don't need to remember these names. These precursors go through metabolic modifications to form other compounds. Okay? And that's how you get all the variety of carotenoid colors. I'll give you a cool example of that. There's a bird called the scarlet minivet, which many of you will probably know from India. The male is red and the female is basically yellow. Alright? Now what happens is, the female they eat the same precursors. The female takes these precursors and deposits them directly into her feathers. The male actually does a little metabolic trick, which is sex specific, and converts them into a red compound that makes his feathers red. So even within sexes of the same species, you can have differences in how carotenoids are metabolized. And that is how they provide the diversity of reds, oranges, and pinks that you see in the bird world. The other major class of pigments in the bird world are melanins. Okay? Melanins, as you know, are compounds that we possess too. They're what give our hair and skin their colors. And melanins are valuable photoprotectors. They protect you against UV light. They are valuable antioxidants, as are carotenoids, which is why these pigments are so important to birds. And they can be used, therefore, to color feathers. And the most important thing about melanins is they can be patterned. So if you see a feather with patterns, bars, flecks, mottling, that's probably a melanin. Okay? And melanins come in a number of forms normally, but the two most common melanins in the bird world are eumelanin. So eumelanin gives birds feathers. They're black, grey and other colors. And you have pheomelanin, PHAEO, pheomelanin, which gives birds light browns, ochres and rust colors. And you can have eumelanin and pheomelanin in ratios in a feather to give it a whole range of melanin pigmented colors, all the way from black to pale yellow brown. And that is very important to remember, because if you actually look at a dark brown or a rust brown color, that is typically caused by a melanin and not a carotenoid. But deep maroons can be caused by a carotenoid. So how do you tell these apart? That's how. Okay? You take the tools that you have biochemically and tell them apart. Are these the only pigments known in birds? No, but they are by far the most common ones. Some examples though. Parrots possess a whole range of yellows and reds, etc. They don't actually have any of these compounds. They don't have carotenoids anyway. They actually possess their own set of compounds called cetacofulvins. Okay? You don't need to know what that is in any detail, but basically remember that parrots have special compounds. In most birds, green is not formed by a pigment. I'll come to green in a minute. But green, in one group of birds, actually comes from a pigment. These are the African turacos. Okay? The turacos are one of the few birds that have a green pigment, and those pigments are porphyrins. Okay? They're called turacoverdins, never mind. They're porphyrins just like hemoglobin. Hemoglobin itself is a pigment that some birds use to color their eyes. And there are a number of other such, mostly minor pigments that are found in specific areas of the body. But by and large, bird feathers are typically colored by either carotenoids or melanins. You'll notice again that I told you to hold on for a minute when I talked about green, and I didn't talk to you at all about blues, violets, and ultraviolet colors. That is because, as I mentioned earlier, birds don't possess pigments for those colors, with the exception of the turacos for green. By and large, blues and violets are obtained through a completely different mechanism in feathers. Right? They are referred to as what are called structural colors. And structural colors can come about through a number of different mechanisms. Alright? But broadly, they are based on one very simple principle. If you have groups of objects arranged in a specific order, when light falls on them, they will function like a diffraction grating and scatter light. And if they are arranged in a specific way, they won't scatter all wavelengths of light equally. As a result, some wavelengths of light will be scattered more than others, giving that substrate a particular color. For that to happen, all you need to remember is you have to have two materials of different refractive indices next to each other. So there has to be an interface, because the scattering happens at the interface of materials of two different refractive indices. If you disrupt this boundary, the scattering will fail and the color will be lost. Alright? In most birds, that interface is, so feathers are made out of keratin, beta keratin, right? Just like our nails and our hair. The interface between beta keratin and a melanin granule that's called a melanosome is one such place where scattering can occur. The other is the interface between beta keratin and air. And I'm going to talk to you about both of those today. Right? How do we know that these scatterings actually occur? One way to do it is these structures are really small, they're microscopic. You take little sections of the feather and look at a transmission electron micrograph. You can look at that and identify periodic structures. Then you can use computational tools to estimate what wavelength of light it's going to scatter. And you can actually mimic these scattering patterns to show that these are in fact scatterers. Okay? They scatter through a number of phenomena which may be familiar to students of physics. Thin film diffraction, scattering by Bragg's law. So if you have crystal-like structures, they scatter using Bragg's law. There's a reason I've not written these down on the board because the math is unimportant here. It is just important to know that if you find ordering, typically your light is scattering coherently. So some wavelengths will scatter coherently and give the feather its color. That's all you need to know. Let's go into some examples of how this might be achieved. Thanks to studies that use TEM, we now have a number of examples of how structural colors are achieved. Okay, let's go into some of them. As I mentioned, feathers, if you remember from other classes, have barbs which then have barbules. So scattering structures can be present either in the barbs or the barbules. Alright? And they can take a number of forms. In the barbules, typically the scattering is between keratin and melanosomes. Alright? And the melanosome arrangement can be either laminar, and I've drawn an approximate laminar arrangement here. One such example is the superb sunbird of Africa that has laminar arrangements such that they scatter light like a thin film of oil on a water surface. This is what you call thin film diffraction. That's why oil on water appears colorful. The same phenomenon happens here. The second arrangement you can have is you can have melanosomes arranged very ordered like a crystal. So you have a square or a hexagonal array of melanosomes that scatter light like a crystal according to Bragg's law. Alright? So the spacing between scatterers will determine what color you get because of Bragg's law. An example of this is a bird called the velvet asceti in Madagascar. Okay? The examples are unimportant, just to know that there are many ways in which you can achieve coherent scattering of light. There is also now a lot more evidence of a form called quasi-ordered. Quasi-ordered, and you can look up the literature on this, means that if you look under a microscope, there doesn't appear to be any order. It seems very random. Okay? But actually there is order, it's just that it is higher order, so you can't really pick it up. And when you look at this higher order structure, it actually still scatters light coherently. So, quasi-ordered structures are found in, for instance, some parrots like lovebirds and others have this sort of arrangement of scatterers. Now here, I've talked to you about how the scatterers are arranged at the microscale. And if they are arranged throughout the barbule, they scatter light uniformly in all directions. As a result, for instance, if you look at a kingfisher's back, it will appear blue no matter where you're viewing it from. Alright? That's because the scatterers scatter light in all directions. Not all feathers do that. Hummingbirds, for instance, have arrays that are not uniform. They are arranged directionally. As a result, what you get is scattering only in some directions, but not others. I hope you see where I'm going with this. If you're standing in the path where it's being scattered, you might see an intense blue color. But if you're off that direction, all you'll see is black. Because of melanosomes, you'll just see black. That kind of highly directional scattering is referred to as iridescence. I'm sure I don't need to tell you what iridescence is. If any of you have purple sunbirds in your backyard, go and look at them and you'll understand exactly what I'm talking about. So remember, all of these can result in either iridescent or non-iridescent structural colors, depending on how the scatterers are arranged within the feather. As I mentioned earlier, you can also have scattering structures in the barbs, in the larger barbs of the feathers. And there, obviously, the scatterers have to be much larger because the barbs are larger. So you can't have melanosomes there. There, typically, you have air. So air structures interspersed with keratin serve as the scatterers here. You can have bubbles of air in the keratin so that it looks like Swiss cheese. That's what barbets, moths, and many other birds have. Or you can have a more ordered arrangement where you have bars of keratin with air bubbles forming interconnected networks with each other, like so, with connections of air bubbles. Many parrots have this, and that's why they are able to exhibit overall uniform bluish colors. Alright? You can see that in many parrots, in fact. Now, so far, as I mentioned, I've only talked to you about scattering structures in the feathers. But those are obviously not the only parts of birds that are colored. I mentioned that the Egyptian vulture is colored in its facial skin using carotenoids. Many birds can have carotenoids in their skin as well. They can flush their skin with blood so that the hemoglobin gives it a red color. I'm sure I don't need to tell you any examples of birds that do that. You probably know quite a few yourself. But skin can also be structurally colored. And that's important to remember. Now, obviously, skin has to have a light scatterer in it to get these structural colors. Now, that can't be a melanosome, and it certainly can't be air. None of us have air bubbles within our skin. But we have collagen fibers. And collagen fibers can form diffraction gratings to cause scattering and therefore blue and green colors in the skin. Again, remember, green is a very special case because it is only in turacos do you have a green pigment. Otherwise, it is always light scattering or another phenomenon that I'll get to in a minute. The eyes of many birds are also colored. And that coloring happens because of crystals in the iris that serve as scatterers. For instance, pterin crystals or guanine crystals can serve as light scattering structures to color irises blue or bright white or any of the other colors that you see in bird irises. And the last thing I want to leave you with is the idea, I hope I've given you a sense, that birds not only have a diversity in their visual systems, but they have a diverse array of colors that come from different mechanisms. So if I were to give you a color and say what is likely to cause it, it's probably one of the three main classes. And how might you find out what is causing the color? You could do investigations biochemically to find out what's causing it. You could measure the reflectance of the color. You could also use a TEM and look at both the barbules and the barbs and explain what sorts of scatterers you might find. But that's what I want to take away. Want you to take away in terms of what sorts of colors birds exhibit. But not all colors are as exclusive as this. And I'll take you to the example of the budgerigar, which is a common pet bird in many households. The wild-type budgerigar is green, but as many of you who own them might know, you also have blue budgerigars and yellow budgerigars. And that is because the green of the budgerigar is a combination of a structural blue color and a pigment psittacofulvin that gives the budgerigar a yellow color. If you mutate the feathers such that they don't deposit melanosomes in them, you lose this and you get a yellow bird. And that's why all the black markings also disappear in the yellow bird. If you mutate them on the other hand so that they can't deposit pigment in their feathers, you get a bird that retains the black markings but is otherwise blue. And the combination of blue and yellow is what gives the green of the wild-type budgerigar. It is a combinatorial color. And that is the last dimension I want to leave you with. If you, for instance, get an assignment that birds have different kinds of mutations giving rise to different colors, that's your answer. That's how to think about it, and that's how to design an experiment around it. And with that, I'll end my class on bird color and leave you guys to go and read more. Please look at the readings that your TAs will prescribe you and try to learn from there a little bit more about the experimental approaches used. I've not gone into great detail since this is still a beginner's class, but for those of you interested in biochemistry, there's a huge literature on this. For our physics and engineering students, there's a lot of literature on newer and more fancy tools that you can use to quantify structural colors. Please go look them up, and as always, ask if there are any questions that you may have. Thank you.